Okay, we started renal physiology with those two codes. So these were renal anatomy was discussed. What are the major points which we discussed that should be noted? At least the terms which will come over and over again. It is the renal cortex, renal medulla, renal pyramid. No face looks like they have ever gone through it once again, at least once more. Any one face? No face. I think those who performed well in the exam, they are not there in the class. They were there in last class, now they are not in this class. Okay, they will copy it out by themselves. Nijera pore nebe, that's okay. But you have to perform in the exam. What is the renal pyramid? Any idea what is the renal pyramid? You are late in the next class. You didn't attend, you were late. But you have not read. Roll 147. What is the renal pyramid? Jode jode. Pyramidal structures, no? Pyramid to jinistari naam. What is the shape? Not triangular. That is called cone-like structures. Cone-like structures whose base is at the seat, whose base is at the, okay, more or less okay, cone-like structures whose base is at the corticomedullary junction and opens into the minor calyx. Now, what are the minor calyx, major calyx? What is the renal pelvis, renal sinus? These were the points to note in anatomy. Then renal lobe and renal lobule. Lobe is, what is the lobe? এইটা এরকম হচ্ছে না, যে লেখাগুলো উপরে চলে আসছে। What is the renal lobe? Renal pyramid which is associated overlying cortex. So whatever pyramid is a part of the lobe but with its cortex above and the minor calyx below. That is the lobe. Whatever she told. Most are looking blank because you have not revised, not even gone through even once, not even looked at the book about renal physiology after the last day's class. So that's why you are looking blank. So I have nothing to do over there. So renal lobe versus renal lobule. The major artery supplying the kidney. So what is the renal artery? Is a branch of the direct lateral branch of the abdominal aorta, right? That is from your anatomy knowledge. I may be wrong, but you have to be right. Then it branches into interlobar, that means between the two lobes. Interlobar to arcuate. As they go round the pyramid, the base of the pyramid at the corticomedullary junctions, it becomes the arcuate artery. Then into the interlobular, and from there the different branches ultimately into the afferent arteriole around the glomerulus, which is the cup-like structure. Glomerulus and the Byman's capsule. The afferent arteriole will form the glomerular capillaries. Some of the blood will be drained into the Byman's capsule. It is not drained. That word drained is wrong. It is filtered, and the rest of the blood will go to the efferent arteriole. For the efferent arteriole, from the efferent arteriole to the peritubular capillaries. Now, there are some nephrons. Nephron is the functional unit of kidney. How many nephrons? Almost around 1 million, 1 to 1.2 million per kidney. So 2.5 million. So what are the nephrons? So these nephrons, some are called superficial nephrons, some are called juxtamedullary nephrons. What we discussed the earlier day. Juxtamedullary nephrons are those nephrons whose these corpuscle, what is renal corpuscle? Glomerulus, Bowman's capsule along with the mesangial cells in the glomerulus. This is the structure of the renal corpuscle. Meaning of the word corpuscle is small rounded structures and not simply cells as we call them in hematology. It is red blood corpuscle, white blood corpuscle, but they are literally, English literature says corpuscles are small rounded structures. So this is a nephron is a very tiny thing. So many in both kidneys. Kidneys weigh 150 gram, almost 115 to 170, 115 to 170, average 150 gram. So 2 kidneys together 300 grams, 2 kidneys together containing 2 million, 2.5 million nephrons. So they are tiny structures. So small rounded body is called a corpuscle. Renal corpuscle is the glomerulus and the surrounding Weimann's capsule along with the intraglomeralular mesangial cells. We discussed up to this. So what will be the blood supply? The blood, this arrangement of the arterioles and capillaries, that is the blood supply. So the renal, interlobar artery, interlobular artery, veins will be accompanying. In case of superficial glomeruli, after the glomerular capillaries are coming out, now they are forming the efferent arteriole, they are forming the peritubular capillaries. Peritubular means around the renal tubule of the nephron, proximal tubule, this part, tubule part, not the glomerulus, not the corpuscle part. Tubule part of the nephron, they are forming the peritubular capillaries. In case of superficial nephrons, the corpuscle is at the above, in the upper portion of the outer side of the cortex. Cortex is itself the outer side, medulla is the inner side. Outer side of the cortex are the superficial nephrons, the peritubular capillaries are dense. And in case of the medullary, the juxtamedullary nephrons, what are the juxtamedullary nephrons? Who else can answer? What are the juxtamedullary nephrons? What do the light fell on you, sorry. The juxtamedullary nephrons, where do the corpuscles lie? This glomerulus, it lies in the lower, at the corticomedullary, near the corticomedullary junctions. That's why juxta means near. This juxta means near, this word I forgot to mention. So everything you have to understand. No way you should memorize without understanding the word. What is the meaning of the word juxta? It is near. So near what? Near the medulla means near the corticomedullary junction. So they start there, and the peritubular capillaries here after the efferent arterioles are coming out, they continue as long straight vessels called the vasa recta. They go deep. So these are the vasa recta. In case of juxtamedullary nephrons, these are the vasa recta, which are surrounding the tubules. Here the tubules are long and the vasa recta are accompanying them. This is the structure in case of juxtamedullary nephrons. What is the percentage of superficial nephrons? More or less 90%. Only 10% are juxtamedullary, or even more than that. So every 9 out of 10 nephrons or 10 out of 11 nephrons are superficial. Rest are juxtamedullary. So here you can't read. You can't read by yourself. These have become small. I read it for you. What are the parts of the tubules? Be it any type of nephron. What are the parts of the tubules? First is proximal convoluted tubule. Then a part of the proximal tubule is straight. Proximal straight tubule. Then it's the thick descending loop of Nenli. We wrote it as DTL. The abbreviations vary in every book to book. You can make your own abbreviation, but you have to understand. During the exam, you don't write abbreviations. At least in the initial part of your answer, you have to write the full term, then write in bracket abbreviations, then you may continue. Better to write whatever DTL, then ATL and TAL. ATL is ascending thin limb. Between the DTL and the ATL is the loop, actual loop proper, where it is the curve proper, this curve. Follow the red dot, this curve. So everything is loop of Henle. This straight part, the straight part of the proximal tubule, then the descending thin limb, then the ascending thin limb including the curve, then the ascending thick limb. Ascending thick limb is written as TAL. So if ascending thin limb ATL, so that is now written as thick ascending TAL. In some textbooks, in other textbooks the abbreviation may vary. So you have to do it, whatever textbook you read, you have to follow the abbreviations. First they will write in one part and they will go on writing the abbreviations. You can do it in your answer script also. You don't need to write the full term every time. Then after the thick ascending, it's the distal tubule. So distal tubule doesn't usually have a straight part. I have not found in textbooks. It is the distal convoluted tubule, either abbreviated either DCT or DT, same thing. Then the then the connecting tubule. At least in one major textbook I have found connecting tubule, then initial collecting tubule, and then the collecting duct. Collecting ducts are not parts of nephrons. So few nephrons up to the connecting tubule. Collecting tubule is part of nephron. Tubule, the term tubule. Then collecting ducts. Few nephrons will go into one collecting duct. This, wherever this is contained, few nephrons, that is the lobule. And many lobules will form a lobe. How many pyramids? 8 to 18 per kidney. Now this is the parts of the tubules. So in the case of the juxtamedullary nephron, tubules are long, along with vasa recta they are accompanying them. In parts of the superficial nephron, the tubules are short and very, so one nephron lies behind another nephron. The density microscopically also very difficult. So to see, so the tubular capillaries, peritubular capillaries are very dense. The efferent arteriole followed by the peritubular capillaries. So see, they are showing whatever we have discussed just now. This is the part. Now one thing is labeled here as the macula densa, which was not discussed last day. Macula densa is the part of a thick ascending limb near the own afferent arteriole. So it is going back, tubule is going back. So loop of Henle, so this is the afferent arteriole near the thick, this is the afferent, this is the efferent. So afferent coming to the capillaries, then the blood is draining by efferent. Near its own afferent arteriole and efferent arteriole, near the own glomerulus of the same nephron, the thick ascending limb is going up. Just where the distal tubule is going to start, the part there is called the macula densa. It is near its, the part is near the glomerulus of the same nephron. So there is the macula densa. That part is called the macula densa. There are some special types of cells there which can sense sodium chloride, the amount of sodium chloride, absorbs how much is coming there, it can sense. So if the amount of sodium chloride is more, then it will influence some other cells, then it will influence something. So we are coming to that. Then it will do something. If the amount of sodium chloride is less, it will do something. So they as if they are acting as chemoreceptors. So macula densa, and we spoke of the mesangial cells which are within the mesangial cells plus matrix. So there are some intraglomerular mesangial cells within the glomerular capillary, some extraglomerular walls. Extraglomerular mesangial cells are also called the lacis cells. Mesangial cells secrete a mesangial matrix containing all these things, collagen, laminin, forget, fibronectin, etc. But mesangial cells are actually supporting cells, but they have other functions also. Mainly they can be called supporting cells. They can be intraglomerular, that is within the capillary there may be mesangial cells. Outside, interspersed between the two capillaries. Capillaries are small, so it is just for, you have to think, imagine it, because microscopically these are very small structures. Imagine how tiny a glomerular capillary can be. Between that there are cells. So mesangial cell, they can be extraglomerular or intraglomerular. Okay, so those are the mesangial cells. Then there is the macula densa, and there is the juxtaglomerular cells. Now what is juxtaglomerular cells? Glomerular begins, glomerulus, by glomerulus we mean mainly the glomerular capillaries along with the mesangial cells. Now there are another types of cells called the juxtaglomerular cells. So juxta means near, likewise juxtamedullary nephron. Now juxta means near. Okay, juxta means near, so juxtaglomerular means near the glomerulus, but it is not within the glomerulus, right? Can you follow? Juxtaglomerular cells you will not be within, mesangial cells can be within. So some mesangial cells are within the capillaries itself, and some that means blood is going within the blood, spots in the blood. The word floating about should not be used, but it is within the blood, within the glomerular capillaries. And some mesangial cells are between, say, two glomerular capillaries, between 2-3 interspersed. Like interstitial space is in between the cells, interstitial space, like that. Mesangial cells, they are extraglomerular, but that means not within the capillaries but interspersed between. And juxtaglomerular cells are not inside this glomerular structure at all, not in the capillary, obviously, not even in between the cells. They are outside, where they are located in the walls of the afferent arteriole, but nearing the glomerulus, in the walls of the afferent arteriole, they are specialized myoepithelial cells. So this tubule, whole tubule is an epithelial structure. It is derived from the epithelium. So we are hearing from the school days, connective tissue is a mesodermal structure and all. What is developed from epithelium? What is epiderm, endoderm, mesoderm, all things. So what is, some structures in the body are developed from the epithelium, some from etc. like this. So tubules is epithelium. So that I think has been discussed in general physiology. So basic things, some basic things you have to remember and conceptualize. So this is epithelial structures means developed from the epithelium. Here you see these I have, forget this B part, don't concentrate B part, concentrate just on the A part. Just to again make it clear where the macula densa, location of the macula densa. Macula densa, okay. So it is the part of the, just where the distal tubule is starting, part of the ascending thick limb, or just part of the initial part of distal tubule, which is near its own glomerulus of the that particular nephron only. So that is the macula densa. These are the mesangial cells, and this is a very looking very complex actually. So these are the extraglomerular mesangial cells. Follow the red dot. Whenever I am speaking there and using this, please follow the red dot only, not the whole thing. Whole thing you can't understand, whole thing, everything is not needed for you also. So this is the embryological development. Very, I require 2 days to study it. So don't go for, you have to don't need these things. Everything you don't need. Wherever I am just pointing, these are the extraglomerular mesangial cells, and these are from very nice textbooks, but you don't need everything. So macula densa, but you need these things. These things you need, whatever I am speaking out. Macula densa, mesangial cells and juxtaglomerular cells. Now what will form the juxtaglomerular apparatus? You have it. Separate competence is there. Discussion about juxtaglomerular apparatus and role of renin-angiotensin system. This is a competency number PY7.2. So you need, what is the juxtaglomerular apparatus? It is the macula densa plus the mesangial cells plus the juxtaglomerular cells. Juxtaglomerular cells are not located in the glomerulus but in the walls of the afferent arteriole. These are myoepithelial cells which secrete renin, and sometimes some books, so these are varying, they are telling erythropoietin is also being secreted from that. Now extraglomerular mesangial cells function, we told, mainly supporting cells, but in times of need they can also secrete renin and erythropoietin. When there is a high amount of renin and erythropoietin required, they can also even secrete renin, the mesangial cells. Together the juxtaglomerular cells, the macula densa and the extramesangial cells constitute the juxtaglomerular apparatus. As if when they are speaking, some terms you will be finding somewhat misnomer. Although we called extraglomerular mesangial cells are interspersed between the glomerular capillaries, so they should not be part of juxtaglomerular apparatus. Why? Because juxta means near but not within. But extramesangial cells are within the glomerulus, still it is part of juxta. So some bit of misnomers you have to accept. Physiological curve in the simple muscle curve. Okay, some misnomers we have to accept. They were named likewise then. So they are now considering glomerulus is just the capillary. So extraglomerular mesangial cells they are considering, while defining the term JGA, they are considering those are outside the glomerulus. Macula densa obviously outside the glomerulus but near to it. It is the part of the distal tubule or the ascending thick limb near to the that particular nephron's glomerulus. So it is near but not within. The juxtaglomerular cells itself are obviously not within, it is within the afferent arteriole, and the extraglomerular mesangial cells, although they are interspersed between the glomerular capillaries, they are now considering it as the part of the juxtaglomerular apparatus. So these types of cells, so JGA will act as part of JGA. Juxtaglomerular cells which are within the afferent arteriole act as baroreceptors. They sense changes in pressure. So can contract. If the myoepithelial cells contract, then blood flow will reduce through the afferent arteriole and into the glomerular capillaries. They act as baroreceptors. The macula densa cells act like chemoreceptors, sensing NaCl into the last part of the ascending thick limb and the first part of the distal tubule. That is the act as chemoreceptors, and mesangial cells act as support, and in times of high needs they can secrete renin and erythropoietin as well. So whatever books you go through, what is the JGA, what are the different types of, what are the substructures, structures in the JGA, what are the functions of those, right? JGA. And what is renin being converted to by the angiotensin converting enzyme 1. I think you have gone through all these things, because aldosterone has also been taught. If you have not gone through, okay, renin angiotensin 1, then angiotensin 2, angiotensin 1 to 2 is by angiotensin converting enzyme, and ultimately to aldosterone. What is the function of aldosterone? Yes, madam, what is the function of aldosterone? Aldosterone is a hormone secreted, it is a hormone secreted from secreted from which part? It is from the cortex of which gland? Please be loud and clear. If you are wrong, but be loud. Adrenal cortex. So next, adrenal cortex secretes aldosterone. What is the function? One part of the adrenal cortex. Again, there are layers. Zona. So, what is the function of aldosterone? Vasoconstriction. Aldosterone. No, very sorry. Aldosterone function don't know. Bolo. Blood pressure regulation. How? সে তো অনেক কিছু থেকেই, বডিতে সব কিছু থেকেই সব কিছু হচ্ছে, ব্লাড প্রেশার রেগুলেশন হচ্ছে, কিন্তু অ্যালডোস্টেরন করছেটা কি? অ্যালডোস্টেরন ব্লাড প্রেশার রেগুলেশন হতে পারে, সেটা পরে গিয়ে। ব্লাড প্রেশার রেগুলেশনের একটা হরমোনের নাম বলতে বললে অ্যালডোস্টেরন বলবে না, তার অন্য হরমোন আছে। হ্যাঁ, রিঅ্যাবজর্পশন, সিট যাই বলছো বসে বলো। রিঅ্যাবজর্পশন অফ সোডিয়াম অ্যান্ড ওয়াটার from ADH acts on collecting duct, aldosterone acts on distal tubule. আর কিছু বলবে? হ্যাঁ, পোটেন্সি। হ্যাঁ, সোডিয়াম ক্লোরাইড রিঅ্যাবজর্বড হয়, তাই ইন এক্সচেঞ্জ পটাশিয়াম বেরিয়ে যায়। পটাশিয়াম ইজ, sodium and chloride and reabsorbs, and potassium is excreted. So that is indirect. So it is sodium chloride reabsorption, sodium and water. Ebar water reabsorption hole fluid volume বাড়বে, blood pressure বাড়বে কি বাড়বে না সেটা secondary. So first is aldosterone and OTA function. And this is not a class on adrenal cortex, but very why I pointed it out, because today is 3rd June. 10th June. 10th June, Tuesday 10th June, and you are appearing for 3rd internal assessment. If classes will be up to say 14th August, 15th August holiday, 16th August, 18th August is your exam. So 2 months of classes more, and on 9th October you are appearing. So very bad if you 4 people, 5 people, ultimately the 6th man could answer the function of aldosterone. And I hope honestly, sir, honestly, you did not see the book? Okay. So he answered honestly, he could, that means he could without referring to any book, text or notes. 6th man. So very bad. Time is being wasted actually. So aldosterone function this. So renin, angiotensin, aldosterone. What is the, now the, how this works? Angiotensin, most potent vasoconstrictor. What vasoconstrictors you have known of? Secreted from vasoconstrictors from the adrenal medulla. Yes, roll 90. Names of vasoconstrictors, names of hormones of adrenal medulla. Sit. Hormones of the adrenal medulla of the adrenal gland, that means inner part. Epinephrine and, so if you say epinephrine, next comes, so epinephrine and norepinephrine. So more potent vasoconstrictor than norepinephrine is angiotensin itself. Then angiotensin, what are the other functions? Sorry, angiotensin 2, renin, angiotensin 1, angiotensin 2, from there aldosterone. So most potent vasoconstrictor angiotensin 2. Then it will help to other functions are, it acts on proximal convoluted tubule for sodium chloride reabsorption. It acts on circumventricular organs in the brain. If special circulation has been discussed along with cardiovascular physiology, so Even if you leave those, you have to write the primary functions. It will raise blood pressure in whatever method, wherever it acts on the body, it will raise blood pressure, increase fluid volume and all. So it helps in the secretion of aldosterone for sodium chloride reabsorption. It acts on the PCT itself. It has a role in acid-base balance also. You will get these functions in the books, whatever books you read point by point. And it not only helps in the secretion of aldosterone, but also secretion of ADH and ACTH. ADH from posterior pituitary, ACTH from anterior pituitary. So these are the different functions, but main is it is the most potent, say the most potent vasoconstrictor, and it will be converted to aldosterone. So this is the Renin-Angiotensin-Aldosterone system, RAAS. So RAAS system, if it comes as a short note, you have to write whatever, 5 marks. So what is your practice in the exam? Hey, tu na? What is your practice in the exam? 5 marks or 15 marks, I write whatever I know. This is the approach by the students mostly here, what we have seen. That should not be the approach. Exam is not for write whatever you know. Exam is for answering the particular question. Questions will be long, questions will be clinical. Okay, we can't afford to wait. Even discussing the exam pattern is a wastage of time now. Renal physiology will need 8 or 9 classes. So up to 14th August, say one class in the week will be the renal physiology. I am trying to cope it up in less number of time because CNS is left, special senses. Although the exam is in October, we have to take the exam in August. That is our limitation. The dates are set by the university such that long before the puja vacation, they will have to submit the marks. The college has to submit the internal assessment marks. So if at least if you got full August, then it would be beneficial. But only up to 14th August. So again, these have been discussed. Okay, renal corpuscle, if you can read the things, it is okay. All these things are shown. Renal corpuscle again is the glomerulus, Bowman's capsule. How is the Bowman's capsule formed? Now Bowman's capsule, we have to discuss 1 or 2 things. What are the filtering layer of the blood. So from the blood, the glomerular capillaries, it will drain into the Bowman's capsule which is surrounding it. What is the Bowman's capsule? It is between the space. Bowman's capsule or space is between capsule, and the space is the space within the visceral and parietal. As in you have read in the lungs and all, there is an outer layer, there is an inner layer. So parietal pleura, visceral pleura, like that. Parietal layer, visceral layer. The visceral layer is composed of podocytes. So podocytes are structures, podocytes are cells, and from them long food processes are coming out. Here are the podocytes. What are the labeled? Podocytes. So this is showing the podocytes. So around the glomerular capillary, podocytes with pedicels, PEDICELS. We are so short of time, it is not possible for me to write everything on the board. Podocytes with pedicels, refer to the books, PEDICELS, food processes of the podocytes. And what is the filtering layer? First will be the glycocalyx layer, even before the endothelium. Endothelium proper, glycocalyx. So glycocalyx is, from your biochemistry knowledge, you should know better than me. Glycoproteins, glycolipids, everything, glycoproteins. So it is a chemical, this is a non-living, glycocalyx layer. Then the endothelium proper, then the basement membrane, then the podocytes, along with the pedicels. This is the filtering layer. Endothelial membrane can't filter large proteins. Proteins will pass through the endothelial membrane, but they will get these negatively charged proteins like albumin. Does albumin usually pass into urine? No. Where will they caught? At the basement membrane, at the podocytes, but not the endothelial layer allows the proteins. That means there are endothelial layer, the fenestrations are not so small that they can catch the larger molecules. So there are the sizes also, greater than 1 kilodalton, you don't have to remember. So you don't need actually. So you are not postgraduates, you are undergraduates. So filtering layer, glycocalyx, then the endothelial membrane, outside that the podocytes, endothelial membrane, then the endothelium, sorry, not the endothelial membrane, endothelium, then the basement membrane, and then the podocytes. So as if the podocytes are the part of the visceral layer of the Bowman's capsule, and then the fluid is draining into the space, and then there is the parietal layer of the Bowman's capsule. If we see a glomerulus, where is the glomerulus? এটাই ছোট হয়ে গেছে। So এই যে, so the visceral, the Bowman space is continuing into the proximal tubule. The space is continuous with the proximal. Which part of the proximal tubule? Proximal convoluted part. Proximal straight part will come later than that. So this Bowman space is continuing in the proximal tubule. But proximal tubules are epithelial in origin. So we have spoke of the juxtaglomerular apparatus, renin-angiotensin system, and basic functional anatomy. If we go on discussing, it can take 10, even 5, 10, 6, 10 classes, including the embryological development, the mesenchymal development, whatever was there in this picture, loose mesenchyme, condensation, how these parts are being formed. Okay. So see, this is urinary space. So this is the space, Bowman's capsule. So this is glomerular capillary lumen, glycocalyx, endothelium, slits within the fenestrations within the endothelium that cannot catch the large proteins. They will pass to, now the glomerular basement membrane will catch. And podocytes, there are spaces within the podocytes also. There will be smaller spaces, but there has to be spaces. Otherwise, how will this filtration occur? So this is the layers of the filtration. So function and anatomy took 1.5 classes. Okay. And it will take, because if I just repeat just one word one time, you can't follow anything, and you are not revising. So that is another thing. After you are not revising, you are again forgetting, but I have to do my job. You are not revising anything. 2 or 3 people are answering, and you have forgot the function of, many of you have forgot the function of aldosterone. It cannot go on like this. So, but I have to do my job. I know it will not be fruitful, but I have to do my job for most of the people. Teachers and classes and complementary. আমি বারবার বলেছি এটা। Teachers, I mean teaching and reading the textbooks are complementary. So একটা করে আরেকটা করব না, যে কোনো একটা না করলেই সেটাই ক্ষতিকর। তোমরা গিয়ে পড়ছো না, সময় পাচ্ছ না, কিছু করার নেই। মেডিকেল ইজ ভেরি টাফ। ফার্স্ট ইয়ার এন্ড ফাইনাল ইয়ার এবং এখানে যেমন বলছি যে জাক্সটাগ্লোমেরুলার এর মধ্যে এটা কর্টেক্স, তার মধ্যে আবার আউটার পার্টস। কর্টেক্স নিজেই আউটার পার্ট, তার আবার আউটার পার্টে সুপারফিশিয়াল নেফ্রন। এইভাবে যেমন মেডিকেল পুরোটাই টাফ। সো ইট উইল গেট ডিফিকাল্ট ইফ আই গো অন টু দ্য নেক্সট থিং উইদাউট রিপিটিং। জানি না কিভাবে কি শেষ হবে, কিন্তু চেষ্টা করে যেতে হবে। সো দিস আর দ্য মেসেঞ্জিয়াল সেলস, লিভ ইট। দিস ইজ দ্য ম্যাকুলা ডেন্সা। নাও পার্টস অফ দ্য just one thing is left in the anatomy. What are the types of cells? So in the tubules, types of cells in the different parts of the tubules. Proximal tubule, inside the lumen. So tubule, there will be the luminar side and the basal side. Basal side means the side towards the peritubular capillaries. Consider the loop of Henle. Consider the tubule. So inner side lumen through which the filtrate is passing now. Ultimately the urine will be formed. This filtrate will ultimately form the urine after reabsorption of substances, maybe secretion of substances. So lumen is carrying, we cannot call it the urine at this point, it is the filtrate. Lumen is carrying the glomerular filtrate, proximal convoluted, carrying the just filtrated glomerular filtrate. So that is the luminar side, and there is the basal side. On the basal side and outside the membrane, there is the peritubular capillaries, vasa extra in case of the juxtamedullary, and simply the peritubular capillaries in case of the superficial, or sometimes there some books are writing cortical nephrons. You should tell superficial, because everything is cortical. Even juxtamedullary nephrons in truest sense of term, they are cortical. It is in the corticomedullary junction near the medulla, but it is cortical. But cortical nephrons, if they write cortical, you have to take it as superficial nephrons. But if you having a choice, we should tell superficial and juxtamedullary. Literally both are cortical. Now proximal convoluted tubule, so they will be having on the basal side many mitochondria, invaginations and mitochondria, and towards the luminar side brush border. Brush border is not contained in any other part of the tubule except the proximal tubule, but all through what is contained, one cilia, cilium, cilium, cilium. Cilium structure you have known from the respiratory system is also discussed, cilium. So it beats in one direction. So all these are for the propulsion of the fluid. Brush border, also you have heard these terms in the intestine. These are the structural features affecting the function. Okay, invaginations and very dense mitochondria. That means many invaginations on the basal side plus mitochondria. All these you will find in the books. Okay, along with many mitochondria. But in the next part, thick limb, thin limb, whatever, thin limb, ascending, descending, next part, what we forgot to mention today, I mentioned the last day, thin ascending limb, see, this green part, follow the red dot and the green line, this green line is the thin ascending limb. In superficial nephron, in juxtamedullary nephron, thin ascending limb is more or less, that is, it can be seen, but in, that means it is a total length of the tubule, thin ascending limb is a significant part. But in case of juxtamedullary nephron, thin ascending limb is very, very short. One book is writing there is no thin ascending limb in the juxtamedullary nephron. This minute part, you follow the change in the color, this green part is the thin ascending limb, it is not, as if it is not there, very small part considering the length in the superficial nephrons. Okay. So that I forgot to mention today, although mentioned last day. So whatever, thick ascending, thin ascending, thin descending and all, thin descending, thin ascending, not many mitochondria and invaginations. Cilium will be there, but not many mitochondria. Distal convoluted tubule, same. Again, distal convoluted tubule, collecting ducts and all, collecting tubules, there will be again invaginations and more mitochondria. Whatever is, so more reabsorption, more, so proximal tubule gets everything. Now it has to do, glucose is mainly reabsorbed in proximal tubule. This will come later, reabsorption will come maybe next day, maybe next class. So wherever there will be the work of the tubules, this reabsorption secretion is more, it will require more energy, mitochondria, ATP. So along like that, the cells are arranged. And what is in the initial collecting tubule, there are two other types of cells, collecting tubule. They are called the principal cells and the intercalated cells. Principal cells are again for reabsorption and all. 2/3 of the cells of this collecting tubule, and intercalated cells, they are regulating the acid-base balance. So one type of intercalated cells, there is a subtype of intercalated cells. Intercalated cells, some terms you have to remember, they can be explained also, but sometimes it is better to remember intercalated cells. So one subtype of intercalated cells secrete H plus in exchange of K plus, that means hydrogen exchange of potassium. Another type secretes bicarbonate, like this. So intercalated cells are more regulating, concerned with acid base balance function, and principal cells with this reabsorption and all. So they are in the collecting tubule. What is cortical collecting tubule? Again, then collecting duct, parts of the collecting duct, outer medullary collecting duct, inner medullary collecting duct. In the large diagram, where was it? So this is the collecting duct. If this is a juxtamedullary nephron, if this is a juxtamedullary nephron, this red dot, then this is the distal convoluted tubule, connecting tubule, initial collecting, going into the collecting duct. One collecting duct will not collect, filtrate from one nephron only. It will collect, filtrate. Now it is nearing urine from many nephrons, forming, and these structure together will form lobe or lobule. One collecting duct collecting from many nephrons will be the lobule. Again, so this collecting duct, this is the outer medullary collecting duct, this is the inner medullary. Medulla is itself inner part. The outer part of medulla is outer medulla, inner part is inner medulla. So superficial nephrons do not reach the inner medulla, but juxtamedullary nephrons reach the inner medulla. So we are repeating it so many times. After this, you have to do it by your own. We have discussed juxtaglomerular apparatus, just macula densa, mesangial cells. Granular cells are the juxtaglomerular cells. Juxtaglomerular cells, they contain granules. These granules contain the renin, which is secreted. Those are the juxtaglomerular cells in the afferent arteriole, myoepithelial cells. Renin-Angiotensin-Aldosterone system. Read the functions of angiotensin. Most important is the most potent vasoconstrictor, even more potent than norepinephrine. And at this point, you are not being able to tell the hormones of the adrenal medulla. So now, one very basic thing: how is the urine formed? Amount filtered. So filtration. After that, there will be reabsorption of substances from the different parts of the tubule. So filtered minus reabsorption. Whatever will be reabsorption is not reabsorbed will not be a part of the urine. এই যে কি ব্যাপার সব বুঝে গেছো? Whatever will be reabsorbed will not be part of the urine. So amount filtered, this is very basic. Easiest slide still now. Amount filtered minus amount reabsorbed. Now from the peritubular capillaries, from the vasa recta arteries, vasa recta, in case of the juxtamedullary, from the peritubular, they are the peritubular capillaries of the juxtamedullary nephron, superficial juxtamedullary, whatever, from the peritubular capillaries, things will be secreted. Things are absorbed, things are secreted. So whatever is reabsorbed into the peritubular capillaries will not be a part of the urine. Whatever will be secreted will be the part of the urine. So ultimately excreted in urine is, so amount of any substance, say glucose. Does glucose appear in urine normally? No. Know this, we will discuss this again. We will discuss this obviously. Tubular reabsorption, not today, next day. But amount filtered minus amount reabsorbed plus amount secreted is amount ultimately excreted. Whatever creatinine is a measurement of renal functions we do. We will discuss, even in your syllabus you have the basic ideas of renal function test, you have to study in the first year only. So creatinine is a marker of renal function. In the laboratories we measure creatinine. That prescriptions we are writing in the OPDs or in the IPDs, prescriptions for a patient, measure of serum creatinine, like complete blood count, total count of say RBCs, total count of WBCs. One is serum creatinine. Serum creatinine measurement is a marker of renal function. So amount filtered. So any substance, it will be amount filtered minus reabsorbed plus secreted is equal to the final amount excreted. Now the concept of renal clearance. Now very important concept once again. I had a plan to discuss many things more. I think this will be the last if I can complete it. Renal clearance is, so if I just have 2 apples in my pocket, any example you take, any stupid example even. I have 2 mobiles. From tomorrow, I bring 1 mobile. Don't think I am very rich, I am very poor, but still I have 2 mobiles. Why? I have a lesson from lockdown, early part of April 2020. My mobile got it. I was very lucky I had a second that I was not using. It was kept in the almirah. But if it was not there, then I had to would have to stay without a mobile for 2 months. So that's why even being very poor, I use 2 mobiles. Because it is no more, this thing is no more a luxury. This is a part of everyday life. Okay. So if one is gone, till repair of that, in the lockdown there was no chance to repair that. So from tomorrow, if I become more poor and I have to sell this to make a living, then I'll have 1. So now I have cleared myself of how much? 50% of this thing. Cleared myself. So how we measure it? It is the amount. It is the amount of that substance. Renal clearance should ideally, we should speak of it as, so 50 milligrams of creatinine have been cleared in the urine. So we should tell creatinine clearance is 50 milligram, maybe per day. I am just taking, just now I spoke 50 milligram, it is not an actual figure, it is a very rough figure. It is a wrong figure actually. 2 milligram, 3 whatever. So 2 micrograms, 5 micrograms, 5 grams, 5 kgs, whatever. We should tell creatinine 5 kgs, or if my body contains 10 kg creatinine and 5 kg has been cleared in a day, I should call 50% of the creatinine has been got rid of another. Now you can obviously understand creatinine will be again formed from muscle creatine. So in the body it is not like that. that I have 2 mobiles, I cannot make more now, I cannot buy at this point more, 3 more. But in the body, substances are being formed and being broken down and going out. But if 50 milligram out of 100 milligram is going out, we should say 50%. Or clearance is 50 milligram per day. But we do not say like that. What we say is the renal, the renal clearance of the substance is the volume of plasma that is completely cleared of the substance. If it is 50 milligram thing, substance is being cleared, but it is from 50, we have to measure the volume of the fluid which contains the 50 milligram. Not measure, we express it like this. So what is the renal, how much blood is flowing through this afferent arterial glomerular capillaries and renal afferent arterial, this blood flow through the kidneys per minute? For now, you know, I will come to this slide. Renal blood flow we will discuss. It is, we discussed last day, it is huge compared to other organs. It is 1.2 to 1.3 liters per minute. Per 100 gram of tissue per minute, it is 300 to 400 milliliter per 100 gram, sorry, how much it will be? 1.2 by 3. 300 to 400 milliliter per 100 gram of tissue per minute. Huge blood flow. But will it clear all the substance from all these 700 milliliter? No. So say sodium. Sodium 700 milliliter is containing lots of sodium. Lots means compared to some other standard. Lots means it does not contain, like I am saying, roughly 1 kg, not 1 kg. It is considered in millimole. Say 1 millimole. But out of that, in one single time, only 0.14 millimole will be cleared. Now, if 700 contains this much, 0.14 will be content will be, I am telling rough figures, but still as far as I remember, in 1 milliliter. 1 milliliter, out of that 700 milliliter, this is 700 milliliter is the plasma, 1.2 liter is the blood. Out of blood, 55% is plasma. Out of 700 milliliter, the amount of sodium cleared is only, say per minute, it is only contained in how much? It is contained in 1 milliliter. Do you get the concept? Now go and read. Say in per minute, 0.2 millimole of sodium is cleared. It is contained in how much fluid or renal plasma? It is 1 milliliter. So clearance of sodium we do not say 0.12 millimole, we say it 1 milliliter per minute, because whatever that is contained in how much plasma, that is the concept of clearance. Why we can learn, but this is the concept of clearance you know for now. So see, how will substances, any substance which has entered through the renal artery, how will they go out? Either it will go through the efferent arterioles into the peritubular capillaries, or to the tubule, reabsorb through the peritubular capillaries, then it will go into the renal vein and again pass out into the blood. Most of the sodium is going back into the blood through the renal vein to the body, but some is excreted through urine. So whatever is excreted through that urine is a millimole or milligram, whatever, but it is contained in how much fluid. That is the concept of renal clearance. So just note this down, is the volume of plasma that is completely cleared of the substance by the kidneys in unit time, and will continue next day.